सो आज हम पढ़ेंगे मेडिकल फिजियोलॉजी, कॉम्प्रिहेंसिव टेक्स्ट बुक ऑफ मेडिकल फिजियोलॉजी वॉल्यूम 1 बाय जीके पाल, चैप्टर नंबर 4। चैप्टर नंबर 4 हमारे लिए हो जाएगा चैप्टर नंबर 4, पेज नंबर आपका 15। चैप्टर का नाम है सेलुलर ऑर्गेनाइजेशन, सेल की फंक्शन एंड इंटरसेलुलर कनेक्शन। कॉम्प्रिहेंसिव टेक्स्ट बुक ऑफ मेडिकल फिजियोलॉजी बाय जीके पाल। ओके, describe the structure and function of a mammalian cell. The cells is the structural and functional unit of tissue and organ of the body. There are about 30 से लेकर के 37 ट्रिलियन सेल in human beings. They provide structural support to the body, utilize nutrient to provide energy, contain body's hereditary code and perform many special function. ठीक है जी। Organization and function of cell. A cell consists of 3 fundamental components: cell membrane, cytoplasm, nucleus. Cell membrane is the protective sheath of the cell. It exchanges material between the cytoplasm and the external environment, facilitate interaction between cell and helps in anchoring the cell to each other. Structure of a cell membrane. The cell membrane is basically a double layer of a lipid molecule having a thickness of 7 to 10 nanometer, into which are inserted or attached various protein molecule. Figure 4.2. Lipid constitute about 42% of the dry weight of the membrane. Protein constitute about 55% and carbohydrate constitute 3%. Fluid mosaic model of the membrane. Though many models for cell membrane have been described in the past, the widely accepted one is fluid mosaic model described by Singer and Nicholson in 1972. ओके, इसको बाद में देखूंगा। स्ट्रक्चर ऑफ सेल, नोट द ऑर्गेनाइजेशन ऑफ वेरियस ऑर्गेनेल्स एंड न्यूक्लियस इन द सेल, बाद में देखेंगे इसको। Special feature of this model are as follows. Cell membrane consists of a double layer of a phospholipid that contains protein molecules. The model is called fluid mosaic model as the membrane lipid are present. The model is called fluid mosaic model as the membrane lipid are present in fluid form that allows the flexibility of the membrane without disturbing the structural integrity. The membrane protein are loosely attached and float in the fluid phospholipid bilayer. Rapid and random redistribution of integral protein occur in the membrane. Also, phospholipid undergoes rapid redistribution in the plane of the membrane. Structure of a membrane fluid mosaic model. Note the arrangement of a lipid bilayer and a protein in the membrane. Okay. This type of a diffusion within the plane of the membrane is called translational diffusion. This occurs rapidly for phospholipid, which can move several micrometer per second. Factor contributing to fluidity of a membrane. The fluidity of a membrane is mainly dependent on the temperature and lipid composition of the membrane. Effect of a temperature. When temperature increases, the hydrophobic side chain undergoes a transition from its ordered gel or crystalline state to a more disordered liquid state. Lipid composition of the membrane. Fluidity is largely dependent on lipid composition. Phospholipid in membrane generally contain at least one unsaturated fatty acid with cis bond. With rise in temperature, the unsaturated bond among fatty acid side chain lose their orderly stiff arrangement. In addition to this, their cis configuration decreases its compactness and become more disordered. Thus, rise in temperature increases fluidity. Similarly, when length of a fatty acid molecule increases, it increases the chance of intermolecular interaction and adds to the rigidity. Thus, with increase in degree of unsaturation, increase in fatty acid length, and increase in phospholipid content containing unsaturated fatty acid and cis configuration membrane and in cis configuration. So phospholipid content membrane fluidity increases. Role of a cholesterol. Though cholesterol profoundly influences membrane fluidity, it behaves differently at different temperature. It is rather a bulky molecule placed among phospholipid bilayer. At increased temperature, with rise in temperature, phospholipid molecule gain kinetic energy and start moving apart. This leads to membrane to start melting, but the presence of a bulky cholesterol molecules pulls the fatty acid together, thus decreasing fluidity. Thus, at increased temperature, cholesterol decreases fluidity of the membrane. At a decreased temperature, when temperature goes down, fatty acid of a phospholipid lose energy and come very close to each other. This leads to decreased intermolecular interaction, which makes the membrane more compact. But the presence of some bulky cholesterol prevent this close interaction, thus increasing fluidity. Thus, at decreased temperature, cholesterol increases membrane fluidity. Thus, cholesterol behaves like a buffer and tries to oppose the effect of a fatty acid on fluidity, thereby tries to preserve the membrane integrity within the range. Hence, cholesterol phospholipid ratio is the important factor for membrane integrity, which in turn is linked to various functions in the cell. Lipid bilayer. Lipid bilayer. The major lipid in cell membrane are phospholipid, glycolipid, and cholesterol. The phospholipid are phosphatidylcholine, sphingomyelin, phosphatidylserine, phosphatidylethanolamine. Glycolipid are generally found in the outer layer. Lipid are amphipathic. Amphi means both. Amphipathic molecule as their head or polar region is hydrophilic and tail, a non-polar region, is hydrophobic. Figure 4.3. The globular or the head end contains phosphate or hydroxyl moiety that are positively charged and soluble in water. The tail end contains two chains of fatty acid that are insoluble in water. The arrangement is such that the hydrophobic tail end are directed towards the center and the hydrophilic head is located to the periphery of the membrane as depicted in figure 4.2. Thus, hydrophilic head of a lipid molecule faces the aqueous phase from both inside and outside of the cell. A similar bilayer arrangement is found in bile salt where they form spherical micelles. Cholesterol. Cholesterol is incorporated into the hydrophobic region of the membrane and serve to reinforce the lipid permeability barrier. The individual phospholipid molecule can move freely within the specific layer but not from one layer to another layer. Hence, the cell membrane is said to be fluid in nature. Application box 4.1. Determinant of a fluidity of a membrane. The fluidity of a membrane depends on the composition of a lipid and degree of unsaturation. The major determinant is its cholesterol phospholipid ratio. Cholesterol at decreased temperature increases the fluidity of the membrane. Function of a lipid bilayer. The main function of a lipid bilayer of a cell membrane is to create a permeability barrier between the interstitial fluid and the cytoplasm. The permeability of a substance depends on whether it is lipid soluble or water soluble. Lipid soluble substance like आपका oxygen, alcohol can pass easily through the cell membrane. Water soluble substance like urea, glucose cannot pass easily. Thus, lipid bilayer makes the membrane semi-permeable. अच्छा, इस चित्र को तो हमें करना ही पड़ेगा. यह amphipathic nature of a membrane lipid. Note that polar head is hydrophilic, non-polar tail is hydrophobic. ठीक ଅଛି, ଏଇଟା ଗୋଟେ ରହିଲା. ତା ତଳେ ଇଏ ରହିଲା, ଏଠି ଗୋଟେ ରହିଲା, ଏଠୁ ଦିଟା କଲମ, ଏମିତି ଆସୁଛି. दिस इज आपका कोलिन, ये होता है आपका फॉस्फेट। ये हो आपका ग्लिसरॉल। ये क्या करता है, फॉर्मिंग द हेड। ये तीनों मिलकर के, कोलिन, फॉस्फेट और ग्लिसरॉल, ये तीन मिलकर के आपका हेड पोर्शन बनाते हैं, फॉर्मिंग द हेड। तापर फैटी एसिड फॉर्मिंग द टेल। दीज आर फैटी एसिड forming the tails. Then membrane protein. Membrane protein are of two type: integral protein, peripheral protein. Figure 4.4. Integral protein ये सब्जेक्ट केते बाटा रही, बाप रे बाप रे बाप रे, ये तो बहुत बड़ा सब्जेक्ट है। 1 2 3 4 5 6, बाप रे। और ये हो गया है कि 7 अध्याय, 15 पेज, बाप रे बाप रे। Integral protein. Some membrane protein that spans entire thickness of the membrane are known as integral protein. They are also called transmembrane protein. Some integral protein penetrate only a portion of the membrane so that they are exposed either to the external environment of the cell, जिसे हम इंटरस्टिशियल फ्लूइड कहते हैं, और टू द साइटोप्लाज्मिक सरफेस ऑफ द मेम्ब्रेन, जिसे हम इंट्रासेलुलर फ्लूइड कहते हैं। Some membrane protein also move laterally within the membrane. Example, membrane receptor can move to the site of endocytosis. The function of integral membrane protein are as follows. Serve as channel protein. Channels or pores are integral protein through which water-soluble substance like glucose electrolyte can diffuse across the cell membrane. Act as a barrier. Carrier protein transport substance through cell membrane by facilitated diffusion. For example, the transport of glucose through glucose transporter. Serve as ion pumps. Membrane proteins serve as pump for active transport of ion across the membrane. For example, sodium potassium ATPase that pumps potassium into the cell and sodium out of the cell against their concentration gradient. Serve as receptor and enzyme protein. Integral protein that are present towards the outer half of the membrane usually serve as receptor, and usually those on the inner half serve as enzyme. Membrane G protein belongs to the category of a protein antigenic function. Complex membrane proteins such as glycoprotein act as antigen on the surface of the cell. For example, the blood group antigen in the membrane of red cell. Peripheral protein. Some protein molecules are inserted lightly in the outer or inner border of the membrane or are just bound to the surface of the membrane. Such proteins are called peripheral protein. There are two types, intrinsic, extrinsic protein. Intrinsic protein they are present on the inner surface of the membrane. They usually serve as enzyme or anchor protein for cytoskeleton and other microfilament that maintain cell shape. Extrinsic protein they are present on the outer surface of the membrane. They serve as cell adhesion molecule CAM for anchoring cell with basal lamina and with neighboring cell. They can be removed without disrupting the membrane. Then membrane carbohydrate. The external surface of the cell membrane is loosely covered by a carbohydrate layer known as the cell coat or glycocalyx. Function of glycocalyx. Glycocalyx serve as a protective coat. Glycocalyx serve as a protective coat. Carbohydrate is negatively charged and therefore prevent negative particles like protein molecule to interact between cells. Some of the transmembrane glycoprotein like selectin recognize and bind with specific oligosaccharide on other cell membrane. Therefore, permit temporary cell-to-cell adhesion. Such temporary adhesion occurs between neutrophil and endothelial cell at the site of inflammation. Stronger adhesion between the cell is formed by integral membrane protein such as integrin. Some carbohydrate molecules serve as receptor. Function of cell membrane. Cell membrane maintain a constant and distinctive intracellular environment which is essential for functioning of the organelle. For example, the intracellular fluid has lower concentration of sodium and chloride, low pH, but higher concentration of potassium, organic phosphate, and magnesium. Cell membrane maintains cell volume by actively transferring ion across it, especially by pumping sodium ion out of the cell. In neuron and muscle cell, it maintains a potential difference between intracellular and extracellular surfaces, which enable cells to respond to various stimuli. Cell membrane helps in recognizing foreign cell or antigen so that they can be destroyed by phagocyte, special feature of RBC membrane. The red cell membrane is most extensively studied membrane. In addition to the general feature of the membrane described above, red cell membrane contains some special protein such as integral and peripheral protein. যেটা সারি না যেটা সারিবি Figure 4.5 Structure of red cell membrane. Note the presence of a special protein such as ankyrin, spectrin, adenosine that provide plasticity or deformability to the membrane. In addition to the presence of a anion exchanger, band 3 protein and glycophorin C and other submembrane protein. अच्छा इसको फिगर को बाद में देखते हैं. Integral protein. Two special type of integral protein are found in red cell membrane. Figure 4.5. These are glycophorin and band 3 protein. बैंड 3 प्रोटीन मतलब एनायन एक्सचेंजर. Glycophorin are glycoprotein that contains 60% carbohydrate and 40% protein. The carbohydrate component is oligosaccharide and the polypeptide component contain 131 amino acid. Oligosaccharide of glycophorin serve as antigen for Mn blood group. Band 3 protein. Band 3 protein is a dimeric protein with molecular weight 93,000 that traverses membrane about 12 times. It acts as a pore or transport protein that exchange bicarbonate ion in the capillaries of lungs. Peripheral protein. The inner surface of a red cell membrane contains two special proteins that are linked to the cytoskeleton and are essential for stabilization of the membrane and biconcave shape of the cell. These are spectrin and ankyrin. Spectrin is a fibrous protein that contains an alpha chain with molecular weight of 2,40,000 and a beta chain with molecular weight 2,20,000. Along with ankyrin, it is attached to the cytoskeletal protein that maintain membrane integrity and cell shape. Ankyrin. The cytoskeletal protein are attached to spectrin through ankyrin. Ankyrin has molecular weight of 2 lakhs. It has two domains, one bind with spectrin and other with N-terminal region of a band 3 protein that extend into the cytoskeleton. Application box 4.2. Disease due to the membrane protein defect. Disease due to the membrane protein defect. Disorder of red cell such as hereditary Spherocytosis and elliptocytosis occurs due to defect in membrane protein. For detail, refer red blood cell. रेड ब्लड सेल, कहाँ पर रेड ब्लड सेल? यहाँ पर कुछ पढ़ा था क्या? ओके. Cytoplasm and cell organelle. Cytoplasm is the body of a cell which is made up of a jelly-like fluid known as cytosol. Dispersed in the cytosol are many particles and organelles. The particles are neutral fat globule, glycogen granules, secretory vesicle, and dissolved protein, electrolyte, and glucose. The usually occurring organelles in animal cell are mitochondria, endoplasmic reticulum, Golgi apparatus, ribosome, peroxisome, lysosome, centrioles. The red blood cell is the only living cell which loses nucleus and most of its organelles such as mitochondria, ribosome, and lysosome during maturation. Mitochondria are the powerhouse of the cell, as with them cell extract energy from the nutrients, and without them cellular function cease. They are cigar-shaped organelles whose shape, size, and number vary in different tissues of the body. They are most abundant in cells and that have high rate of metabolism as in liver, cardiac muscle, etc. Mitochondria are distributed in all parts of the cytoplasm, though they are concentrated more in those portions of the cell that have major share of its energy metabolism. Total number of mitochondria per cell varies from less than 100 up to several thousand, depending on the metabolic activity of the cell and the amount of energy required by the cell. Mitochondria are variable in size and shape. Some are a few hundred nanometer in diameter and globular in shape, whereas others are elongated, having 1 micrometer diameter and 7 micrometer length. Even few mitochondria are branching and filamentous structure. Mitochondria consist of two important components, membrane and cristae. Mitochondrial membrane. Electron microscopy shows that each mitochondrion has two layers of a membrane, the outer and inner membrane. Outer mitochondrial membrane. This forms a continuous envelope of the organelle. Outer membrane consists mostly of phospholipid and cholesterol and contain a specific membrane protein that forms porins. Porins are channels that permit substance with molecular weight of less than 10,000 to diffuse freely across the outer membrane. Inner mitochondrial membrane. It is rich in protein and the ratio of lipid to protein is 0.27 to 1. Hence, it is virtually impermeable to polar and ionic substances. The inner membrane is folded into multiple incomplete septa like structure called as cristae. Figure 4.6 structure of mitochondria. Note that cristae are infolding of the inner mitochondrial membrane. Okay. Which is rich in many enzymes like cytochrome B, C1, C, A, and A3, NADH dehydrogenase, succinate dehydrogenase, electron transferring flavoprotein, carnitine, carnitine, palmitoyl transferase, etc. Important note: number of cristae decreases with activity. Number of cristae is more in resting state of mitochondrion and decreases in respiring state. Mitochondrial matrix. The region enclosed by the inner membrane is called matrix. An amorphous material fills the matrix which contain enzyme involved in Krebs citric acid cycle and fatty acid oxidation. Table 4.1. Matrix also contains several strands of DNA, ribosome, and enzyme for synthesis of a protein coded in the mitochondrial genome. Function: cellular oxidation. The inner membrane contains the cytochrome of the electron transport system and the associated enzyme for oxidative phosphorylation. Mitochondria are the major site of cellular oxidation of a nutrient, formation of ATP. The tissue cycle takes place in the matrix. The initial breakdown of a protein, carbohydrate, and fats occur in the cytoplasm of the cell, and the final end product that enters the mitochondria as acetyl coenzyme A combines with oxaloacetate to form citric acid. The end product of a citric acid cycle, that is Krebs cycle, are CO2, H2O, ATP. In glucose metabolism, glucose is converted to pyruvic acid in the cytoplasm by glycolysis, and during the process 2 ATP are generated. Pyruvic acid is converted to acetyl coenzyme A in the matrix of mitochondria, which then enter tissue cycle that yields 36 ATP molecule. Thus, 95% of a cell's ATP formation occur in the mitochondria. Table 4.1 Mitochondrial enzyme. Number A. Membrane enzyme. Enzyme of outer membrane. cytochrome B5, B5, cytochrome B5 and cytochrome B5 reductase, fatty acid coenzyme A synthase, phospholipase A, nucleoside diphosphokinase। दूसरा होता है एंजाइम ऑफ इनर मेम्ब्रेन, साइटोक्रोम बी, C1, C, A and A3, NADPH dehydrogenase, succinate dehydrogenase, electron transferring flavoprotein, beta OH butyrate dehydrogenase, carnitine palmitoyl transferase, all translocases। देन बी में एंजाइम इन इंटरमेम्ब्रेन स्पेस, adenylate kinase, nucleoside diphosphokinase, sulfite oxidase। देन सी में एंजाइम इन द मैट्रिक्स, पाइरूवेट डिहाइड्रोजिनेज कॉम्प्लेक्स, साइट्रेट सिंथेज, आइसोसाइट्रेट डिहाइड्रोजिनेज, मालेट डिहाइड्रोजिनेज, फैटी एसिड ऑक्सीडेशन सिस्टम, ऑर्निथाइन ट्रांस कार्बामाइलेज, अल्फा ऑक्सोग्लूटारेट डिहाइड्रोजिनेज, एकोनिटेज। Important note: use of ATP for cellular function. ATP are used for three major functions of the cell. Number one, for transport of ion and organic substance through cell membrane. For example, ATP is used mostly for transport of sodium ion, potassium ion, calcium ion, hydrogen ion and magnesium ion, phosphate, glucose, etc. Phosphates, glucose, etc. Number two, for synthesis of chemical compounds such as synthesis of protein, phospholipid, cholesterol, purine, pyrimidine, etc. Number three, for mechanical work such as muscle contraction. Now, number three, function of a mitochondrial matrix. As they have strand of a DNA, they are capable of self-replication as well as protein synthesis. Lifespan of a mitochondria. Like most components of the cell, mitochondria have a short lifespan and are constantly renewed. But as they participate in cell activity and oxidation, they are damaged by oxidant generated in the vicinity. Clinical box 4.1. Mitochondrial disease and dysfunction. The disease that affect mitochondrial energy transduction is called Loft's disease. The abnormality of mitochondrial DNA leads to cellular dysfunction called as mitochondrial cytopathy syndrome, which manifests with muscle weakness, degenerative lesion of the brain, and a high level of lactic acid in the blood. Mitochondria are damaged by free radicals and affected in age-related degeneration, that is oxidative aging. Then coming to the endoplasmic reticulum. Endoplasmic reticulum consists of a network of anastomosing membranous tubules, vesicle, and flattened cisternae. The membrane of endoplasmic reticulum are continuous with the outer membrane of nucleus and are also connected with Golgi apparatus. Figure 4.7. Endoplasmic reticulum is the site of synthesis of protein and lipid for the membrane of cell and organelle and secretory vesicle of the cytoplasm. There are two types, rough endoplasmic reticulum, smooth endoplasmic reticulum. In most cell, rough endoplasmic reticulum and smooth endoplasmic reticulum remain in continuity like two parts of the organelles and work in an integrated manner. Rough or granular endoplasmic reticulum. When surface of endoplasmic reticulum is studied with ribosome, the organelle is called rough endoplasmic reticulum or granular endoplasmic reticulum, as it gives a rough or granular appearance to it. In some cells, such as red blood cell, the ribosome lie free in the cytoplasm. Rough endoplasmic reticulum are present in more number in cells that are actively involved in protein synthesis, like acinar cell of pancreas and neurons. In neuron, the nasal granule are modified rough endoplasmic reticulum. Function of rough endoplasmic reticulum. It is concerned with protein synthesis, therefore it is abundant in cells of endocrine gland and cells secreting digestive enzyme. Protein molecules are synthesized within the structure of a ribosome attached to the surface of endoplasmic reticulum. Protein molecules are extruded from the wall of endoplasmic reticulum to the interior of endoplasmic vesicles, tubules, and matrix. It also plays some role in glycosylation, that is conjugation of a carbohydrate with protein to form glycoprotein, a function which it shares with the Golgi apparatus. Figure 4.7. Structure of endoplasmic reticulum. Note that the inner part of endoplasmic reticulum is in continuity with the nuclear membrane and the outer part interact with the Golgi complex. Rough endoplasmic reticulum has numerous ribosomes on the membrane and smooth endoplasmic reticulum has no ribosome on the surface. Smooth or agranular endoplasmic reticulum. When ribosomes are not attached to the surface of endoplasmic reticulum, the organelles is called smooth endoplasmic reticulum or agranular endoplasmic reticulum, as its surface has smooth or agranular appearance. Function of a smooth endoplasmic reticulum. It is concerned with the synthesis of a lipid, hence it is abundant in cells that synthesize cholesterol, steroid hormone, phospholipid. In muscle, it is called sarcoplasmic reticulum, which is specialized for the storage of a calcium ion that is released during the excitation, contraction, coupling. Smooth endoplasmic reticulum is part of a intracellular transport system as it is continuous with the rough endoplasmic reticulum and Golgi apparatus. It is also the site for detoxification or neutralization of hormone and toxic substances. Important notes. Endoplasmic reticulum vesicles rapidly migrate to Golgi apparatus. Lipid molecules formed are rapidly incorporated into the endoplasmic reticulum that increases the size of endoplasmic reticulum. To keep endoplasmic reticulum growing beyond a limit, a small vesicle called endoplasmic reticulum vesicle or transport vesicle continually break away from smooth endoplasmic reticulum and mostly migrate to Golgi apparatus. Figure 4.7 depicts migration of a vesicle from endoplasmic reticulum to Golgi apparatus. Now coming to the Golgi apparatus. Golgi apparatus. The Golgi apparatus is also known as Golgi complex or dicytosomes. It is present in all cells and generally is located close to the nucleus. It appears as flat discs that are dilated peripherally and stacked together in a bunch of stacks of cisternae. Figure 4.8. Anatomically and functionally, it is closely associated with endoplasmic reticulum. Functionally, Golgi apparatus two main regions are cis and trans. The region near to the nucleus is the cis phase, जिसको हम cis Golgi के नाम से जानते हैं, and the region close to the membrane in the trans phase that is known as trans Golgi. Now, structure of a Golgi complex. Note the structure mainly consists of stacks of cisternae. Now, function of a Golgi apparatus. Formation of a secretory vesicle. Golgi apparatus is the site for packaging of a secretory product into the secretory granules. Material produced in the rough endoplasmic reticulum travels through lumen of a smooth endoplasmic reticulum vesicle budding off from the smooth endoplasmic reticulum transport material to the cis phase of the Golgi complex. Hence, they are also called transport vesicles. Transport vesicle fuse with the Golgi apparatus and empty their content into the Golgi apparatus. Some protein are phosphorylated and glycosylated here. Also, in Golgi apparatus, the secretion from endoplasmic reticulum are compacted into highly concentrated packets. These packets then pass to the trans phase where they are packed into the secretory vesicles. See figure 4.7. Finally, those small and large vesicles continually break away from Golgi apparatus and diffuse into the cytosol or discharge outside as the cell secretion by exocytosis. The process of cell secretion in granular tissue happen in 1 to 2 hours. Important note, process of cell secretion in glandular tissue happens within 2 hours. When a glandular cell is bathed in amino acid tagged with radioactive material, the radioactive protein molecules are detected in endoplasmic reticulum in 5 minutes. In Golgi apparatus within half an hour and are secreted to the cell surface in 1 to 2 hours. Then synthetic function. It is the site for incorporation of a carbohydrate into a newly synthesized protein to form glycoprotein. Certain carbohydrate molecules such as large saccharide polymer cannot be formed in endoplasmic reticulum. These carbohydrates bound with small amount of a protein are synthesized in Golgi apparatus as it occurs in formation of hyaluronic acid and chondroitin sulfate. Chondroitin sulfate. Important notes. Hyaluronic acid and chondroitin sulfate have many functions. Hyaluronic acid and chondroitin sulfate are major component of organic matrix of cartilages and bones. They are major constituent of proteoglycans in the mucus and glandular secretion. They form the ground substance in the extracellular matrix of interstitial tissue space. They facilitate cell function such as migration and proliferation. Now, number 3. Formation of a primary lysosome. Primary lysosome containing lysosomal enzyme are formed in Golgi apparatus. These lysosomal vesicles extruded from the Golgi apparatus are called Golgi hydrolase vesicle. Figure 4.9. Formation of a primary lysosome, secondary lysosome, and phagolysosomes. Now, number 4. Transport of a material. Transport of a material. By discharging material through the vesicle, Golgi apparatus helps in transport of a material to other organelles and to the cell surface. Replenishes cell membrane. Secretory vesicles formed in the Golgi apparatus are discharged to the cell surface by exocytosis. In the process, the membrane of a Golgi apparatus is incorporated into the membrane of the cell. Figure 4.7 depicts the fusion of a membrane of a vesicle with cell membrane during discharge of a secretion by exocytosis. This fusion helps in expanding the cell membrane and replenishing the membrane that is lost or used up for formation of phagosomes during phagocytosis. Thus, cell membrane is continually renewed and replenished by Golgi apparatus. Lysosomes. Lysosomes are membrane-bound spherical organelles that contain a variety of hydrolytic enzymes meant for intracytoplasmic digestion. More than 40 different lysosomal enzymes, that is lysozymes, have been isolated. Some important lysozymes are listed in table number 4.2. Lysozymes are found in almost all animal cells except erythrocyte. They are particularly abundant in cells having high phagocytic activity such as neutrophil and monocyte. Important notes. Absence of lysosomes. Lysosomes are present in all cells except RBC. They are prominently present in neutrophils. In granulocyte, the lysosome appear as cytoplasmic granule. Lysosomal activity of a cell or tissue can be determined During the process of phagocytosis, the phagosomes that is formed in the cytoplasmic pseudopodia containing foreign bodies such as bacteria, virus, etc. fuse with lysosomes to form phagolysosomes. See figure 4.9. And lysosomal enzyme later digest these foreign organisms. Therefore, lysosomes are known as autophagosomes. Application box 4.3. Marker for lysosomal activity. Acid phosphatase is used as a marker for lysosomal activity. Hmm. नहीं. Important lysosomal enzyme. A. प्रोटियोलाइटिक एंजाइम जिसमें कैथेप्सिन, प्रोटीनेजेस, कोलाजिनेज, इलास्टेज. लिपोलाइटिक एंजाइम लाइपेज, फॉस्फोलाइपेज, फैटी एसाइल एस्टरेजेस. Then carbohydrate splitting enzyme alpha glycosidase, beta galactosidase, hyaluronidase, aryl sulfatase. Nucleic acid hydrolyzing enzyme ribonuclease, deoxyribonuclease. Other enzyme में acid phosphatase catalase. The interior of a lysosome is acidic. pH is about 5 compared to its cytosolic exterior. pH of a cytoplasm is about 7.2. Acidic interior of this organelle is due to the action of a proton pump. The PMH plus ATPase present on the lysosomal membrane that pumps proton H plus from the cytosol into the lysosomal interior against its electrochemical gradient. The lysosomal enzymes are acid hydrolysases as they function best in acidic pH. Types of lysosomes. There are three forms of lysosomes. प्राइमरी लाइसोसोम, सेकेंडरी लाइसोसोम जिसे एंडोलाइसोसोम बोला जाता है, एंड टर्शियरी लाइसोसोम जिसे फागोलाइसोसोम बोला जाता है. फ्लो चार्ट 4.1. प्राइमरी लाइसोसोम, दे आर फॉर्म्ड विथ हाइड्रोलाइटिक एंजाइम सिंथेसाइज्ड बाय रफ एंडोप्लाज्मिक रेटिकुलम and packaged in the Golgi apparatus. They are generally referred to as storage vacuoles. Secondary lysosome. These are lysosomes fused with endosomes, hence they are called endolysosomes. Tertiary lysosomes. They are autophagic vacuoles or autophagosomes. They are formed by fusion of phagocytic vacuoles with primary lysosomes. Hence, they are called phagolysosomes. Flowchart 4.1 Types of lysosome and their function. Lysosome तीन प्रकार का है. प्राइमरी लाइसोसोम गोल्गी हाइड्रोलेज वेसिकल, सेकेंडरी लाइसोसोम एंडोलाइसोसोम, टर्शियरी लाइसोसोम फागो लाइसोसोम्स. देन प्राइमरी लाइसोसोम जो होता है वो न्यूली फॉर्म्ड लाइसोसोम, एंजाइम इनएक्टिव. सेकेंडरी लाइसोसोम में प्राइमरी लाइसोसोम फ्यूज्ड विथ एंडोसोम, एंजाइम एक्टिव होता है. टर्शियरी लाइसोसोम contain degraded product after phagocytosis. No enzymatic activity seen. Important note. Residual bodies. After the material in phagosomes have been digested by lysosome, some waste material may be left. The undigestible remnant in the lysosome are called as residual bodies. Residual bodies containing indigestible substance are excreted from the cell by exocytosis. Function of lysosomes. Digestive organ of the cell. Hmm. 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 Function of lysosome. Function of lysosome. Digestive organ of the cell. Lysosome contain many enzymes essential for intracellular digestion. Absence of these enzymes leads to various storage disease. Clinical box 4.2 are listed in uh, hmm, are listed in table 4.3. Bactericidal activity. Lysosome kill and remove ineffective organisms and foreign bodies. Lysosomes are highly bactericidal. Mechanism of killing of a bacteria by lysosome include lysozymes that dissolve cell membrane of the bacteria. Number 1. Number 2. Lysoferrin present in lysosome binds iron and other substance before they can promote bacterial growth. Number 3. Acidic pH of about 5.0 in lysozymes activates lysosomal hydrolysis and inactivate bacterial metabolic system. Number 3. Autolysis. When a cell is damaged or ruptured, the enzyme liberate within the cytoplasm, digest the cytoplasmic content, a process known as autolysis. The autolysis enzyme are mainly lysosomal enzyme. Important note: autolysis has two important functions: regression of tissue. Few tissue in the body regress to a smaller size. For example, regression of uterus after pregnancy, atrophy of muscle during long period of inactivity, and decrease in size of a mammary gland after the period of lactation. Lysosomal enzyme facilitate this process of regression, though the mechanism of increased lysosomal activity in these tissue is not clearly known. Removal of a damaged cell or tissue. During damage to the tissue caused by mechanical, thermal, chemical, or any other injury, lysosomes are ruptured and hydrolyses are released that digest the cell material and surrounding organic matrix. The damaged cells are digested by the process called autolysis and are completely removed. Later, the new cell formed by mitotic reproduction from the adjacent cell replace the old or damaged cell. Table 4.3. Common lysosomal storage disease. डिजीज एंजाइम डेफिशिएंसी यूनिक फीचर फाब्री डिजीज इसमें क्या एंजाइम डेफिशिएंसी होता है अल्फा गैलाक्टोसिडेज ए यूनिक फीचर क्या होता है क्यूटानियस एंजियोकेराटोमास एंड हाइपोहाइड्रोजेस गाउचर डिजीज जिसमें एंजाइम डेफिशिएंसी होता है एसिड बीटा ग्लूकोसिडेज यूनिक फीचर क्या होता है हेपाटोस्प्लिनोमेगाली एंड स्केलेटल डिस्प्लाजिया देन नीमैन पीक डिजीज जिसमें एंजाइम डेफिशिएंसी होता है स्पिंजोमाइलिनेज यूनिक फीचर क्या होता है मेंटल रिटार्डेशन सीजर लंग फेल्योर देन टे साक डिजीज जिसमें एंजाइम डेफिशिएंसी होता है बीटा हेक्सोसामिनिडेज ए जिसमें यूनिक फीचर होता है मेंटल रिटार्डेशन, मैक्रोसेफाली एंड हाइपर एक्यूसिस इन इन्फेंट, हुर्लर डिजीज जिसमें एंजाइम डेफिशिएंसी होता है अल्फा एल इडुरोनिडेज, यूनिक फीचर होता है मेंटल रिटार्डेशन, कोरेज फेसिज, सीवीएस इन्वॉल्वमेंट। देन और एक डिजीज होता है पॉम्पी डिजीज जिसमें एंजाइम डेफिशिएंसी होता है एसिड अल्फा ग्लूकोसिडेज, यूनिक फीचर होता है मायोकार्डियोपैथी। देन फोर्थ फंक्शन ऑफ लाइसोजाइम रो क्या रोची रिसाइक्लिंग ऑफ सेल ऑर्गेनेल्स। The worn out cell organelles are transported to the lysosome by autophagosomes and are then transferred into the interior of lysosome. Lysosomal enzyme digest these organelles and process is known as autophagy. However, in this process, the nutrient liberated from the digested organelle are reused by the cell. This is one among the several mechanisms of cell survival during scarcity of nutrients. Important notes. Autophagy is the housekeeping phenomenon of the cell. ऑटोफैजी मतलब टू ईट वन सेल्फ ऑटोफैजी by lysosome is the process of killing and degrading the degenerating and obsolete organelles of the cell. Also, large protein aggregates are degraded and recycled. Thus, autophagy is a housekeeping process of the cell. Autophagy contributes to routine turnover of the cytoplasmic components. Number 5. Fertilization of ovum. Acrosomes located on the head of a spermatozoa is a specialized lysosome that contains many hydrolytic enzymes. Acrosomes play an important role in penetration of ovum by sperm, removal of a product of a metabolism. Lysosomes remove intracellular product of a metabolism from the cell. Therefore, in the lysosomal storage disease, deficiency of a specific enzyme leads to accumulation of these metabolic products in lysosomes and inclusion bodies in the cell that causes cell dysfunction. Clinical box 4.2. Lysosomal storage disease. Congenital absence of a lysosomal enzyme leads to accumulation of a material in the lysosomes that are usually degraded by the lysozyme, causing lysosomal storage disease, especially elimination of a certain substance from the cell of the body. Example, glycogen, cerebrosidase, gangliosides, and sphingomyelin mainly depend on the activity of a lysosomal enzyme. Therefore, in the absence of a active lysozymes, intracellular accumulation of these substances interfere with normal cell function and produces disease. Accumulation of a partly digested cellular material is called inclusion bodies. Then ribosomes. Though ribosomes are usually present on the surface of endoplasmic reticulum, they are also present as a free organelles in the cytoplasm. They contain about 85% of the RNA of the cell. They are the major site of protein synthesis. RNA is also present in nucleus, mitochondria, cytoplasm, and consequently protein synthesis occur in this structure too. They may be present singly, मतलब monoribosome, or in groups, polyribosome. Then coming to the peroxisomes. Hmm. Peroxisome are the small spherical organelle having diameter about 0.5 micrometer and therefore denoted as microbodies. Specialities of a peroxisome. They are formed by budding from or by division of a smooth endoplasmic reticulum. They are referred to as subcellular respiratory organelles, but they do not have energy coupled electron transport system. They contain oxidases that promote oxidation of lipid, especially omega oxidation of a long chain fatty acid forming acetyl coenzyme A and hydrogen peroxide H2O2 and catalases that liberate oxygen from H2O2. They protect the tissue from oxidative stress. Application box 4.4. Okay. Peroxisomes protect from oxidative stress. OS. Hydrogen, hydrogen peroxide, a potent oxidant produced during metabolic reaction, is one of the agents for oxidative stress. H2O2 is destroyed by catalases, a peroxisomal enzyme which is designated as an antioxidant for combating oxygen oxidative stress. Thus, peroxisomes protect cell from the toxic effect of H2O2. Hence, increased catalase activity is one of the marker of OS. Then number 3. Number 3, specialities of peroxisomes. With the help of a peroxine, the protein chaperone or chaperones, various protein with specific signal sequence are directed to peroxisomes. The membrane of a peroxisome contain a number of specific protein that transport substance between peroxisomes matrix and cytosol. Peroxisome matrix contain more than 50 enzymes that are involved in many metabolic reactions of lipid, long chain fatty acid, plasmalogen, cholesterol, bile acid, purine, amino acid, H2O2. Deficiency of enzyme lead to many diseases. Clinical box 4.3. Proliferation of peroxisome is caused by many synthetic products that act on receptor in the nuclei of the cell known as peroxisome proliferation activated receptor. Peroxisome proliferation activated receptor PPARs are the member of a nuclear receptor superfamily. Those on activation bind to DNA and produce change in mRNA production. Peroxisome PPARs have extensive physiologic effect and affect many tissue and organ. Clinical box 4.3. Disease of paroxysm. Paroxysm may be abnormal or absent in inherited disorder. Zellweger syndrome. Zellweger syndrome occurs due to the mutation in the gene encoding certain proteins such as peroxin and peroxisomal enzyme. This syndrome is characterized by profound neurologic impairment, accumulation of a very long chain fatty acid, abnormalities in synthesis of a bile acid, and marked reduction of a plasmogens. The child usually dies within a year in this disorder. Another similar genetic disorder is infantile Refsum disease, which is less severe and only few proteins are affected. Then Brown Slider disease, adrenoleukodystrophy occurs due to the insufficient oxidation of a very long chain fatty acid by peroxisome. This is an autosomal recessive disease that manifests with progressive degeneration of liver, kidney, and brain. Primary hyperoxaluria occurs due to the defective peroxisomal metabolism of a glycosylate derived from glycine. Then coming to the सेंट्रोजम के ऊपर हम नेक्स्ट में पढ़ेंगे। आज और भी मतलब 3 पेज आई थिंक रह गया हो। 3 पेज, ये एक हाफ पेज, 6-7 टोटल हो जाएगा। तो थैंक्स फॉर वॉचिंग।